We're going to cover the Chapter 14 concept map. What I want you guys to do is follow along and answer the questions in the appropriate spot on the concept map. For example, make sure you guys are able to determine what floor is. Be able to define floor. Also, make sure you guys are able to distinguish between resident floor and transient floor. So make sure you're able to define, provide an example of resident floor, define, and provide an example for transient floor. In addition, make sure you're able to determine factors that are actually causes of why transient bacteria only stays day to day. Be sure to discuss how you acquire your natural floor. There are a few examples that occur throughout your life where you acquire your natural floor. Make sure you guys are able to determine what may cause your bacterial flora to fluctuate. There are a bunch of examples. Please state 4. And then make sure you guys are also able to determine the difference between sterile sites and harbor sites. Harbor sites would be sites that normally house bacteria. So be able to determine a body part that's sterile and possibly a fluid that's sterile, a body part that normally has bacteria, and possibly a fluid that normally has bacteria as well. Next, we're going to talk about factors of infection disease. For example, make sure you guys are able to define pathogenicity and be able to distinguish between a true pathogen and an opportunistic pathogen. So define what a true pathogen is and give an example. And then define what an opportunistic pathogen is and again give an example. Also state what LD50 and ID50 are used for and be able to determine which LD50 or ID50 is more virulent. As far as establishment, make sure you guys are aware of the virulent factors. And there are 4 virulent factors that we talked about. The first factor that we talked about were adhesions. Make sure you're able to define what adhesions actually do and then provide examples. There are a few different examples. One that allows for attachment due to specific receptors. One that allows for attachment due to a sticky factor. And one where the attachment is due to tiny, like hair structures that are able to grab onto and adhere to cell surfaces. Antiphagocytic factors, make sure you guys are able to define what they are, and there are types or examples of them. There is an antiphagocytic factor that actually kills white blood cells. Make sure you're able to determine what that one is. There's also antiphagocytic factors that make the bacteria very slippery and therefore prevent the actual bacteria from being able to be grabbed onto, engulfed, and digested. Extracellular enzymes, there are a few that we talked about. Make sure you guys are able to define or be aware of coagulase and what coagulase does, as well as kinase. Define what those guys do. Toxins, remember, are your poisons. There are 2 types of toxins, exo and endo. Make sure you're able to determine what type of bacteria the exotoxins are found in, gram-positive or negative. Same thing for endotoxin. Make sure you determine where the toxin is found. Is it found in the cytoplasm or the cell wall? And same thing for the endotoxin. Where is the toxin found? Cytoplasm or cell wall. I want you also to be able to tell me if the exotoxin is specific to its target or systemic, as well as the endotoxin. Is it specific to its target or systemic? One of the last topics that we talked about was the disease and spread of disease, termed epidemiology. Make sure you guys are able to define what epidemiology is. Also be able to determine and indicate the different types of graphs. The graph where there are small numbers of cases that are unrelated. The term that refers to where a disease is always present in a population. Where the endemic numbers rise above the norm and then what happens when the epidemic numbers become high and are common among different continents. Be able to distinguish between signs and symptoms. Are signs measurable or subjective, and are symptoms measurable and subjective? And also be able to provide an example of each. Make sure you guys are able to determine an etiological agent, define what that is, and provide an example of an etiological agent. Reservoirs are where pathogens reside normally, and there are 3 different areas. Human reservoirs, be able to define and give me examples of some human reservoirs. Animal reservoirs, what are some bacteria or viruses that are found only in animals? Remember, the other word for animal reservoir is zoonotic. And then what are examples of some non-living reservoirs and some bacteria or viruses that may be found in those non-living reservoirs. Additionally, we talked about modes of transmission. Vehicle are usually non-living factors. Be able to provide examples of vehicle transmission. Vector usually involves insects or bugs, and there are 2 types of vectors, mechanical and biological. Be able to provide examples of those. And then there are 3 types of contact transmission, state what they are, and again provide an example of each. Lastly, make sure you guys are able to determine what are some factors that lead to emerging infections. These are infections that are new to an area. So make sure you're able to provide here examples of the factor as well as the diseases that are associated with them. Make sure you guys are able to define what nosocomial infection is and provide examples of some nosocomial infections as well.